Delhi University South Campus. And he's Dr. Rajiv Kumar Kapoor. He's Senior Assistant Professor at Maharshi Dayananda University in Department of Microbiology, and Department of Biological and Agricultural Engineering at University of Arkansas in USA. He worked for some time on enzyme-based detoxification of fermented inhibitors. And he did his PhD in South Campus, Delhi University, and that is in microbiology itself. And he did his master's and PG diploma, and he's I think patent work is also, he's a consultant for patent work also, and he's associated with various activities of National Institute of Immunology, one of pioneering institute in India, and Institute of Genomics and Integrative Biology. And he's also I told you, qualified patent agent for Indian Patent Office. He's also worked for the Amarsham Pharmacia Biotech Asia Pacific Limited to product specialist, and later genetics. Like these are the people who work for the molecular biology grade components and equipments. He also worked as a project manager in intellectual property division of Eluvera Private Limited, Gurugaon, where they will file the patents, and he was a consultant for them. So he's having both industrial experience, patent experience, and as well as academic experience, and having a good exposure of IIM and Delhi University South Campus. And I know personally because we work together, and with his knowledge, actually he can, because of we need a slot for biochemistry, he can do the justice for anything. We were looking for him, but in future we'll accommodate at different places. So now he will be delivering about biochemistry, isolation of, see, basic DNA estimation, glucose estimation, ethanol estimations, that like any manual will help you to do those things. So the major things like isolations of biomolecules, DNA, RNA, proteins, that will be a challenging task, and their estimations is a big task. So we didn't gone with the basic things, glucose estimation, ethanol estimation. So we have gone with biomolecules, isolation, as well as estimations. So I request Raju to enlighten our participants and us in whatever the assigned areas, along with if the time permits, your research area that you are carrying out at MDU, so that our participant will be enlightened, and they will be encouraged to do the research in the particular fields. Over to Raju.
Right. Thank you, Chan, for those wonderful words for me. I'm really delighted to be present here before you. Hyderabad is one of my favorite cities. Right now I'm sitting in Haryana, Rohtak, and the weather here is rainy, and I can see the rain pouring in down here. So today I'm here before you to acquaint you with the technique of nucleic acid isolation. We will be separately taking DNA followed by RNA, and I will also let you know how to estimate DNA and how to estimate RNA, and we will also touch upon how protein estimation is being carried out. So these are some very basic techniques which lay the foundation for higher techniques like genetic engineering, cloning, sequencing, microarray technology. So understanding this technique is very important so that you can build upon it and you can understand a more sophisticated technique like DNA microarray technology or the next generation sequencing technology. Even cloning and genetic engineering, they all require isolation of DNA. So I'll begin my talk with the first presentation, which is about isolation of DNA. So we'll take up DNA first. I'll share my screen now. So you can see my screen now. the outline is that first, what is the purpose? I just now explained you the purpose of extracting DNA is to, this is the starting point where we
Raju, unmute yourself, please. Devender.
now you
Okay.
can
Yeah,
hear.
that is audible.
Okay, so I think you missed my previous words. Okay, okay. So uh, thank you for taking me in as a speaker for today. I'll begin my talk with the DNA extraction. This is the technique which lays the foundation for many higher techniques, many sophisticated techniques like next generation sequencing, cloning, genetic engineering, microarray technology. Whatever you think about, the first thing which comes to the mind is that isolation of DNA. So this is basic to every sophisticated technique. This technique lays the foundation for all those sophisticated techniques which we are nowadays coming across. So whatever you do, the first step for any genetic engineering process is to isolate uh, DNA, and that DNA should be in the purest form. So the purpose of DNA extraction is to start with, for every technique, just I named few techniques like next generation sequencing, cloning, genetic engineering, microarray, uh, gene therapy, whatever you name, the first thing which comes to the mind is that your DNA should be extracted, and that should be in the purest form. That is very important, that whenever we extract DNA, there are many methods. And if you look into the manuals, there are many methods of isolation of DNA. So, but for every method, there is one or the other variation to optimize the process for isolation of DNA. So today we will see what are the, first we will learn about the basics, and then we will get into the alteration variations where we can vary the process and we can get the DNA in the purest form. So we need DNA in the purest form. So for that, nowadays you will see lot of kits coming in which use some resins which are actually charged resins. These resins are used for isolation of DNA in the purest form. Chilax is the technology which uses charged resins for isolation of DNA. We will briefly learn that also. But the whole idea is to get the DNA in the purest form. Now, let's see what is the basic protocol for the isolation. Now, most uh, most of the DNA extraction processes, it has two parts, major parts. First is a technique to lyse the cells, because the lysis of the cell is very important. Now, the cells can come from different sources. It could be a liver sample, it could be a plant sample, it could be a bacteria. So any living cell, we we can isolate DNA from any living cell. It could be a follicle of your hair. Hair is also a living cell. It could be your nails. We can isolate DNA from nails also. We can isolate DNA from blood. So name any cell, even the fossilized cell. If you want to study the fossils, if we can have a good fossil sample, many times it has been seen that the DNA has not been degraded. So even the fossilized samples, you can have you should know how to isolate DNA from a fossil sample. So the protocol for DNA isolation, it keeps varying with the kind of cell you have. So let me tell you, if it is a plant cell, so in a plant cell, you see a cellulosic cell wall. When it comes to an animal cell, you see a membrane. And when it comes to a bacterial cell, you see a peptidoglycan layer, gram-positive, and gram-positive, it's a lipid layer. So you see the architecture of the cell keeps varying. The architecture of the cell keeps varying. And so your protocol also has to be optimized for every kind of cell. But the basis of the process remains the same. The basis, if I talk about the basis, what I'm going to discuss in the PPT is the basic process, and then we will see how this process is applied to isolation of DNA from bacteria or a plant. So first thing is to lyse the cell. So lysis can be achieved by different means. You can use mechanical shearing. You can use in different chemicals. For example, in case of yeast cells, you use toluene for cell disruption. If it comes to bacteria, you might use lysozyme. You might use proteinase K. Proteinase K is nowadays most frequently used because it is very effective in denaturing the ribonuclease is present in the mixture, and it also digests all the proteins present in the sample. So the proteins present in the sample gets digested, and the cell wall ruptures, and the protoplasm is released. So this is what is the motive of lysing. Sorry. This is what is the motive, first motive, that is the lysing the cell. So you can use enzymatic, chemicals, or mechanical methods for the lysis. So the next, I've explained you about lysis. The next step is precipitation. Precipitation refers to separating phases, or separating a solute from the solvent. So in biochemistry, if you see, how do you achieve precipitation is you all might have read about the technique of salting out. How to disconnect the solutes from the solvent. So this is very important. This is the basis for every precipitation reaction, that how to disconnect a solute from the solvent. How to finish, how to kill the relationship between a solute and a solvent. So that is something which we achieve by using different solvents. We achieve by using different solvents the disconnection of the solute from the solvent. So that is precipitation. In case of DNA isolation, you will see that this is achieved by certain salts. This can be achieved by uh, alcoholic solvents, isopropanol, ethanol. These are the solvents which disconnect or this precipitate, right? Then comes washing. Washing, once you have your DNA, once you precipitate the DNA, there are many other salts which get precipitated along with the DNA. So these salts have to be removed. These salts have to be removed, and the DNA should get rid of the salts and other impurities which get precipitated together. So we we always use washing steps uh, in precipitation. Next is resuspension, and in resuspension, we resuspend the DNA, and the DNA gets resuspended, and this is generally done before you check the DNA uh, on genetic sources. So in this slide, this I've already spoken about. Lysis can be done by liquid nitrogen. Precipitation is phenol chloroform extraction to get rid of proteins. And precipitation can be done by different salts, which is very cheap. Nowadays, precipitation by salts is very cheap to achieve. And then using an ethanol solution, we precipitate. Lastly, washing of the pellet, air drying it, and then dissolving it the tris EDTA buffer, which has preservation properties also. So in DNA extraction, when it comes to the plants, I told you that there is a thick cellulosic membrane. So we always prefer to use liquid nitrogen. I told you that the first step for extraction from any kind of sample is the disruption of the cell, and in case of the plants, this disruption is achieved by grinding in liquid nitrogen. So once you grind in the liquid nitrogen, it causes a lot of mechanical shearing uh, in the cells, and the protoplasm is liberated, and now you have membrane covered cells which are just like protoplasts, and these membrane you can easily get rid of this membrane by using some detergent, which liberates the whole protoplasm into the cell, into your test tube or ependoff, and then later you can precipitate or denature the proteins, which is achieved by phenol chloroform isomyl alcohol. So while you're using the second step after the lysis step is the phenol chloroform isomyl extraction, and the the role of phenol is a denaturing agent, and it denatures all the proteins. It is a deproteinizing agent. So phenol chloroform together, they uh, they uh, they form such kind of a linkage that they deproteinize all the proteins, and then you can see that the proteins get denatured in the form of a whitish thick substance, which you can see in the ependoff. So the moment you add phenol chloroform, it is an organic solvent. It forms a separate phase, but by shaking the ependoff, you make sure that all the cellular milieu, or all the protoplasmic component, which includes your DNA, also comes in contact with the PCI, what we call it, phenol chloroform isomyl alcohol. So once your protoplasm comes in contact with the PCI, the proteins get precipitated or denatured. So the denatured proteins can visibly be seen in the ependoff tubes. And once you do the spin, you can see three phases coming, and these phases, you can see the organic phase and the aqueous phase. So the DNA is in your aqueous phase, and it aqueous phase has to be separated down. So once you separate the aqueous phase, the DNA has to be precipitated using organic solvent. We generally use organic solvents which are having high dielectric constant, which actually kill the relationship between the water and the DNA. So after you precipitate the DNA, the final step is washing and resuspension. So the precipitated DNA is laden with acetate salts, and we use 70% ethanol to remove the salts or other impurities which comes along with the DNA. And the resuspension can be performed in double distilled water or 1xTE buffer. So once the resuspension is done, your sample is ready for to be uploaded onto a gel. Now, this is the overview of the process. First, the lysis, then the removal of membrane lipids, then protein denaturation, which is carried out by phenylchloroform isoamyl alcohol. Uh, uh, then we wash it, and then optionally we give RNase treatment because once you isolate DNA, it is a nucleic acid. RNA is also a nucleic acid,
Sir,
and
sorry for disturbance, sir. Sir, the screen is not visible, sir. Make it bigger size, sir.
okay, all right. So, the because of the presence of the nucleic acid, RNA is considered as a contaminant when we are isolating DNA. So we give a RNase treatment, and then we again elute in slightly alkaline buffer, and we dissolve our DNA in purified double distilled water. So this was the basis of the whole process of DNA isolation. So now, after you do all your studies, uh, let me hide this. Okay, so I'm showing you certain slides. Uh, Yeah. So what are the expected results in a research lab? So you can see here in the gel, we have isolated DNA from different sources. So this gel is a 0.8% agarose gel, and after you isolate your DNA, then you examine your DNA for the integrity of the DNA. So you do gel electrophoresis, and if you see bands which are intact and which should be slightly on the upper side. If it is coming down, that means the quality of the DNA that you have extracted is not good. It has sheared. So shearing causes reduction in the molecular weight of a protein, and the bands shift to downwards, which doesn't indicate a good quality of DNA. So the bands should have some roots also in this picture. You I Let me tell you that these bands are looking good, but the process of the DNA extraction, I see the bands on the lower side. They should be here. If the process is good, you see bands near the well because you're isolating the genomic DNA here from the cell. So your band should not go down. It should not be low molecular weight DNA, because if it is coming like this, that the your DNA is low molecular weight, that means the procedure that you followed caused a lot of shearing of DNA. A good process is what I expect my band to be somewhere here. So, and the band should have certain roots. When it moves downwards, the DNA moves downwards, it shows some trails. I'll show you in my another paper. Let me show you uh, another paper, one of my paper which shows uh, what you see here is one of my research papers. This was my first research paper that is improving the yield and quality of DNA isolated from a fungi sample. This I did it in 2003. So why I'm showing you this is to show you now. Please look at these bands. These are showing some trails which are pointing towards the well. In this picture also, you can see trails pointing towards the well. This indicates the good integrity of your process, because the genomic DNA has to be a high molecular weight DNA, and the high molecular weight DNA always shows trailing trails behind, right? So if you can see such kind of trails or marks and a thick intact band which is on the upper side, which is more than 23.1. This is a Hen3 digested DNA marker. So this is a 23.1 kb, the first band. The DNA band is even above it. So that means you have isolated a good quality of DNA, right? And the roots coming out shows a good quality of DNA isolation. So in this paper, the method was to use CTAP. This is another detergent which we use for isolation of DNA from fungi. In case of bacteria, you see the usage of SDS, sodium dodecyl sulfate. But depending upon the source of your cells, you can change the detergents. So I told you that the basis of all the processes for DNA isolation remains same, but every process has to be optimized. When you are using two different fungi, even you have to optimize the concentration of detergents in two different fungi to obtain high molecular weight DNA. For bacteria, more or less the process remains same, but if when you are isolating from plants and higher organisms, you have to optimize your protocol, and every protocol changes from one lab to another. So I come back to my presentation here. Okay, so now in this is also a common thing that is observed in the labs, that you have a lot of the bands and there's a lot of glow coming down, and in this specifically I wanted to show this because there is one lane here which is showing no bands, but we can see a smear. So what is this smear? So when this smear generally happens when you are not handling your sample properly, and your DNA is getting degraded. So this is the smear which generally you come across when the process of your handling of DNA is not good, and your DNA sample gets degraded. So what is expected out of here is that uh, the DNA from the in the E lane is sheared, broken up into numerous fragments, and is not a clean single band at the top. So these are middle-sized DNA which has given rise to this smear. Okay, so now I will show you a video which will explain the process of DNA isolation in more detail. So now here you see that the in this video you see this, there's the falcon tube. In this falcon tube is we have already grown our uh, cells. Start from the beginning. So the first step is that we have our cells pelleted already. These ependophs are having pelleted cells which have been grown in LB media, the Luria Bertani media, and as a after the pelleting of the cells, we are now adding the uh, lysis buffer into the pellets and resuspending the cell. So the first step for any DNA isolation process is the addition of the lysis buffer. Here you see the lysis buffer is added into the these ependophs, and these ependophs already have in them.
Raju, video is not coming.
Can you see the video now?
Yeah, now it is coming. Yeah,
okay. Yeah. So
Yes,
what
sir.
I was saying that the first step in this process is the pelleting of the cells, and in this ependoph tubes you already see a pellet there. The cells which were grown, overnight, which generally achieve a OD of 0.6 to 0.8, are used for isolation of DNA. And here you see the cells are pelleted. So after the pelleted the cells, we resuspend the cell, which I told you the first step is the lysis. So we add the lysis buffer into the cell pellet and resuspend the cells. Now this lysis buffer, the composition of this lysis buffer varies. I told you in beginning also that whatever is the source of your cell, if it is a bacteria, the lysis buffer will contain lysozyme or proteinase K. If it is if it is a plant cell, it might contain cell degrading enzymes also. So depending upon the source of your cell, you will have different kind of lysis buffer. The other thing which is present in the lysis buffer is the detergent. That is, in case of bacteria, we use SDS, sodium dodecyl sulfate, which actually uh, uh, turns the membrane into mysis. It disrupts the lipid membrane. Now let's move on to the next step. So this is the resuspension. We are resuspending the pellet. You can see the pellet is getting resuspended. Now, what was shown here was the RNase. This was RNase. So RNase is an endonuclease enzyme which is actually added at the time of the lysis, after adding the lysis buffer. This is optional. This can be used at the end of the step towards after the precipitation and resuspension of the DNA also. But in this video, RNase is being added right at the first step after addition of the lysis buffer. So this will ensure that the DNA is pure has a contamination of RNA. So RNase is added at a concentration of 10 mg per ml. This is a commercially available enzyme. After this, the mixture is incubated for 1 hour. You can see this is a water bath which is preheated to 37 degrees Celsius, and we have to incubate our cells which are resuspended in the lysis buffer and we have also added the RNAs to it. Now, after doing that, we are using proteinase K. The function of proteinase K is the digestion of proteins. Uh, it also denatures the uh, nucleases present in the your appendoph. Generally, when the cell gets disrupted, there are certain nucleases also which get liberated into the cellular milieu. So these nucleases cause a lot of problem by degrading the DNA or the nucleic acids. So it is very important to denature these nucleases. Here we are using proteinase K. That is another agent which we use in most of the buffer is that EDTA. EDTA is a chelating agent, and this agent is used for uh, precipitation of chelation of the magnesium ions which are present in your appendoph. So if you know that nucleases are metalloenzymes which need the presence of magnesium for their actions. If you are chelating magnesium from the mixture, the nucleic acids, the nucleases will no longer be able to work and your DNA will be safe. So proteinase K also acts in both the way it denatures uh, the nucleases, it disrupts the proteins, liberating the DNA. Now, incubate after adding proteinase K, you have to incubate overnight, and then next morning come and add phenol chloroform isomyl alcohol. The ratio is 25 is to 24 is to 1. So here the phenol should be tris saturated. The phenol has a very high pH. I don't know whether you checked it any time or not, but phenol is corrosive. It has a very high pH, more than 10, 11. So your DNA cannot tolerate such a high pH. So your phenol has to be tris saturated. What I'm saying tris saturated means you have to add tris buffer into phenol, and then you have to saturate your phenol with tris. Uh, During this process, you will see the pH of your mixture, the PCI, what we call it as PCI, is also comes down from 11, 12 to 7. So now your DNA is safe. When you're giving a phenol chloroform isomyl extraction, your DNA is safe because the phenol is tris saturated. That means its pH has been brought down by addition of the tris buffer. Chloroform, as you know, I have told you, it's a deproteinizing agent, and isomyl alcohol is generally added to stop frothing. It's an anti-frothing agent. So if remember, if there is a lot of frothing in your appendoph tube, your DNA will get entangled in the froth and will be sheared. So you should take care that there should be no frothing in your appendoph tube. You should gently handle the pipette. You should not give jerks to the appendoph tube. So there should be no frothing in your appendoph tube. This is where handling matters. So you see, they are very gently giving the phenol chloroform isomyl alcohol treatment after the lysis buffer has been added. Now we have to centrifuge it, keeping the RPM as 13,000 RPM for 10 minutes. Now this step will separate the organic and the aqueous phase, and you will see that in your appendoph there are clear, you see here, there is a clear aqueous phase, and then here you see some whitish precipitating precipitation, which is actually the protein, and below here, which is hidden by this the fingers, is the organic phase, which is actually phenol chloroform isoamyl alcohol. So your DNA is in the aqueous phase. You have to separate very carefully this aqueous phase without disturbing the precipitant. You because this layer is dynamic, it moves, and if you pull the DNA with a jerk, then you can take away the precipitated protein along with the aqueous phase also. So you have to very carefully remove the aqueous phase. And what I suggest is that in this video, though they are the tip, if you see the tip of the pipette, this should be cut. You can cut the tip slightly so that when your DNA enters through a narrow pipette tip, it gets sheared. If you can cut your tip and make it slightly wider, you can ensure that your DNA doesn't get sheared because if because you have to take this from a narrow pipette. So now your this step can be repeated twice to get a good quality DNA. So we can repeat phenol chloroform isoamyl alcohol and get more pure form of the DNA. Now to the aqueous phase, we are again adding chloroform. You can see in this step, and we are mixing it. This is again to get completely rid of the proteins. Then after addition of chloroform, we are giving a spin for 10 minutes. And then again separating the aqueous phase from the organic phase without disturbing the interface. Actually, the interface is the place where most of the proteins are precipitated proteins are present. So similarly, chloroform treatment can also be repeated to get more. Now we are adding 3 molar of sodium acetate. This is a salt, and I told you that salts are using are precipitating agents. And after adding sodium acetate, then we add 2 volumes of 100% ethanol. So if the volume of your aqueous phase is 100 microliters, add 2 volumes, that is 200 microliters of ethanol. At this step, you will see that the DNA starts precipitating, and if you see the hands, you can see the DNA precipitated, and it just started settling down. So here you have the DNA. You can leave it overnight for precipitation, which will ensure. Then next morning, come give a spin for 30 minutes at 4 degrees Celsius, and then the pellet you have has to be air dried. You remove the supernatant, that is the ethanol, and now air dry the pellet. The pellet may be washed again with 70% ethanol to remove any salts which have precipitated along. So here you see that the pellet is being washed, and again we give a spin. You have to remember that all the spins should be carried out at 4 degrees Celsius to ensure that the DNA doesn't get damaged. Now again remove the supernatant. This you've completed the washing step now. Now you can remove the minor remnants of the ethanol by pipetting, then leave the tubes open for some time to air dry. This step should generally be done in laminar flow because in India we have a lot of air contamination. Now, after air drying, resuspend the pellet in an appropriate volume of TE buffer. Appropriate means a small volume which ensures the resuspension of the pellet. So now your sample is ready for to be loaded onto a gel electrophoresis, and it can be seen as and once you load it on gel electrophoresis, you can see the quality of the DNA that you have isolated by the shape and size of the bands. Of course, if this is a genomic DNA, you should expect the band at the higher side, high molecular weight band. Okay, so now we have finished with the DNA part, and now we are moving to our the next part.
Raju,
Yeah.
try to be a bit louder. Voice is a bit lagging. Maybe the net connection also the problem.
Okay. Okay.
Yeah.
I'll bring the mic more closer.
Yeah. Yeah.
So now let me start with the alkaline lysis, because we are talking about DNA. Actually, a plasmid is also a DNA, and the plasmid are nothing but extra chromosomal elements present in bacterial cells, and plasmids have played a great role in uh, in genetic engineering, because they are the vectors. They're used as a vector for carrying our desired DNA to the host cell. So plasmids are of different types. It could be bacterial plasmid. It can come from different kind of bacteria. And in this process, what we call as alkaline lysis, here we are isolating the plasmid. Now, this is a simple procedure. Uh, we have to make 3 reagents before we start the process. And you can see in the first reagent is a resuspension buffer. That means after pelleting of overnight grown culture, when I'm saying culture, that means this culture is having a plasmid DNA. Generally, we use a DH5 alpha, which is the culture of E. coli, which has a plasmid DNA, which harbors a plasmid, right? So we once we grow the cell overnight, and it achieves an OD of zero point 0.6 to zero point 0.8, we pellet it, and the first step is that we use a resuspension buffer. So this has trisacil, which is a pH 8, glucose, which regulates the osmoticum, EDTA I told you is a chelating agent, which ensures the removal of the metal ions from the mixture, a buffer you're using, and once all the metal ions are removed, nucleases are no longer able to work, and this ensures the intact isolation of your plasmid. Then is the denaturing solution. This is alkali. This is known to lyse the cells, and we are adding SDS, which is known to remove the lipid bilayer from the surface of the cells. Now, the third solution is potassium acetate, right? So you can see the solution 2 has NaOH. That means we are the pH here must be 10 to 11, but in solution 3 the pH is towards the acidic side, 2, 3, 2 or 3. So there is a big difference you might have noted in these two solutions. In solution 2 the pH is alkaline, and in solution 3 the pH is acidic. I'll tell you what difference that does that make. So quickly taking you through the steps, grow the culture overnight on single colonies, which are containing your plasmid. Then take 1.5 ml of these culture, which most of the time is grown in LB media, and then you add it to microfuse tube, then you pellet it at 10,000 RPM for 30 seconds. Remove the supernatant, which is actually the media, and now you have the bacterial pellet. Now you resuspend this bacterial pellet in solution 1. Now, what was the solution 1? It was having glucose, EDTA, and tris HCl. So this is a resuspension buffer, which is actually not harming the cell, which is just ensuring that the cells get surrounded in a proper buffer media. Now it has, you see the solution 1 has tris HCl, pH 8. pH 8 is this a very stable pH for your nucleic acid. So before you lyse the cell, you have to ensure that it is surrounded by a buffer which gives it a proper environment. Because the moment DNA or nucleic acid come out into your tube, into the environment, they should not be getting a shock. So we ensure that the cells are first resuspended in a buffer which gives a same, which is of the same physiological pH which is there inside the cell. And 25 millimolar, this is the ionic strength of this tris buffer, which is same as the ionic strength present inside the cell. So resuspension buffer just ensures that the cells are suspended in a media or in a buffer which is actually the same as inside the cell, because now in the next step the DNA or the nucleic acid, which is the plasmid, and the DNA both will be liberated. So after adding solution 1, vortex, vortexing is for resuspension, then add 200 microliters of solution 2. Now solution 2, you remember it is NaOH, and it is highly alkaline. This will NaOH and SDS. So this solution 2 will actually be disrupting the cell completely, and SDS present in the cell, in the buffer, will remove or will dissolve the bilipid layer which is now exposed. Now, after adding solution 2, you invert the tube very gently. This is very important that you don't vortex it. It has to be very gently inverted 3 to 5 times, and it should be done till you see the solution getting viscous. The viscosity of the solution increases in this step because all the cellular contents are liberated into the buffer, and that makes the buffer viscous. Now, after adding solution 2, incubate it in ice for 5 minutes, then add cold solution 3. Now, you remember the solution 3 was acidic. The solution 3 was acidic, and which the acidic pH was achieved by sodium acetate and acetic acid. Now, after giving alkaline treatment in solution 2, now we are changing the pH to acidic. Now, why this? Because when we add alkali, the DNA is, in the previous step, the DNA is precipitated, and in this precipitated DNA, the plasmid is also entangled, because plasmid is of a smaller size. Generally, plasmid is from 4 to 10 kilobase pair of size, but once when it gets precipitated, it gets entangled in the high molecular weight DNA. So this DNA, now you have to change the pH, which is done by adding an acidic solution. So when the pH is resumed back towards normal, because solution 2 was highly alkaline, now solution 3 is acidic. When you are slowly adding sodium acetate, the DNA starts renaturing. It starts coming up and it gets renatured, and this renatured DNA liberates the small sized plasmid first into the supernatant. So the supernatant has the plasmids, and the high molecular weight genomic DNA, it gets separated out. It is separated out. So that's how once you do this sodium acetate solution 3 treatment, then you incubate it on ice for some time, then centrifuge for 5 minutes at uh, 12,000 G, then collect the supernatant. You can add RNase A to remove all the RNA present in it, which can be there as a contaminant. You can do a phenol chloroform extraction, which will ensure proper removal of the proteins from the plasmid DNA sample. For the supernatant, and these steps are same as we did for the DNA isolation. That is washing off your pellet, then air drying of the pellet, then resuspending the pellet, and 50 microliters of Tris EDTA buffer. Then after that, you should check the quality of your plasmid. Now I took this picture just to show that the gel which will appear, like the gel will be showing more than one band. Plasmid DNA never comes up as a single band. Unless you digest it, it will never come as a single band. So the plasmid DNA, whenever you isolate, you have these many bands, which is showing in this picture. So these bands actually correspond to different forms of plasmid. The plasmid is comes in different form. It can come in a circular form. It can be a linear plasmid also. It could be a supercoiled. You might have seen that as if a rope is made into a circle, you can change it into a supercoil. You can straighten the rope. You can make it into a circle also. Open circular means when the double stranded plasmid is nicked, it becomes open circular. So you never see a single band of a plasmid. Uh, you see multiple bands of a plasmid, which also show a variation. The same plasmid, it is the same plasmid which will move differently in the gel. You have to remember this, that the same plasmid moves differently in the gel because it corresponds to a different form of plasmid. The physical form of the plasmid is different. In the picture here, you see that on the right side, there is only one band. If you want to change this plasmid on the left side, where you see multiple bands, into a single band, you have to digest the plasmid with a restriction enzyme. Then all the forms of the plasmid, they linearize and they give you a single band. But before digestion, after isolation, the plasmid always comes as a multiple band, which actually correspond to different forms of plasmid. So in this picture, this is different forms of plasmid is shown. The same plasmid can you show up as three bands. It could be open circular, it could be linear, it could be supercoiled, it could be circular. So the plasmid could be in any of these three or four forms. And when you isolate the plasmid, you will see these four forms as separate bands. So you should know what these separate bands correspond to and how to get a single band single band can be achieved, because this is the same plasmid which is having in different forms. When you do a restriction digestion, all the plasmid linearizes and you can see a single band as shown in this figure. So this was about my notes on plasmid DNA isolation. Now I will show you a small video uh, which is on plasmid DNA isolation. Now these are the cells. These are cells which have which be E. coli DH5 alpha, which harbor the plasmid. Now these cells have been grown overnight, and we are taking 500 microliters or 100 more than that. Any plasmid DNA isolation is that you have to grow the cell overnight in LB media. Here we have grown the cells on Luria buttoni media for overnight till the OD of the cells is 0.6 to 0.8. Now in this picture you see a test tube in which the cells were grown. You can see the test tube is turbid. That means we have achieved a significant growth of the cells. Now we are transferring 1.5 ml of this culture into a Eppendorf tube. This step is called harvesting, because now after transferring the cells, we will centrifuge this culture so that we can pellet the cells. So this step is done by centrifuging at 13,000 RPM for 2 minutes. Centrifugation is generally, first step centrifugation is to get rid of the media. Here you see the media components have been poured into the drain, and you can see the cell pellets sticking at back of the Eppendorf tube. So we are completely removing the LB media. Now the first buffer, as I told you, was the buffer which is used for resuspension. So this buffer is having SDS, it is having tris SCl pH 8. This is just to resuspend the cells in a suitable media. Now we use the second solution, which is now, this has NaOH and SDS. Now this, you can see how gently this solution is being taken care. We don't have to vortex it. It has to be gently inverted 5 or 6 times. And in this, at this moment, you will see there is a change in the viscosity of the culture, because all the cells are now lysed and all the media is liberated. Everything is now liberated into the mixture, into the Eppendorf tube, that is making the mixture very viscous. Now we add buffer 3, which you remember is the acidic, which is having sodium acetate and acetic acid. This step is there, which will bring the pH towards the normal side now. Now again, we are very gently inverting the tubes at least 5 or 6 times, so that the turbidness is good. And after that, we centrifuge again at the same RPM for 15 minutes. Now you see a supernatant and a whitish thing which is sticking to the Eppendorf. So this here you can see the whitish thing actually represents the high molecular weight DNA and the cell debris, and the supernatant, which is clear, is actually having your plasmid. The whitish thing which is sticking here is the cell debris and the high molecular weight genomic DNA, which is entangled and is not able to completely renature. Actually, when we added solution 3, the pH was shifted from a higher side towards the neutral side. This quickly renatures the small molecular weight DNA, but the high molecular weight DNA still is entangled in the cell debris. So this is how we are pulling out, or we are separating out, the low molecular weight plasmid, which is actually 2 to 8 or 10 kb in size. So you see, we have very carefully removed the clear fluid, and we have left the debris. Now this, in this video, this has been added to a spin column. Now this, in this spin column, just carefully see that there is, yeah. So here you see there is a resin. There is a resin. This is a tube within a tube, and at the bottom of the tube there is a resin, which is a charged resin, which will bind all the nucleic acids. Now, once you add the clear fluid, this fluid has the plasmid DNA. Now, you put this fluid into this smaller tube cartridge, which has a resin and opening. The opening is to allow the fluids, but the nucleic acids will bind to this resin. So this is a proprietary resin. Qiagen was the first to invent it, but nowadays you find many other kits which are using the same resin. This is a charged resin which uses the polyanionic charge over the DNA or the nucleic acids to trap them. Now, after centrifuging this, we centrifuge at 8,000 RPM for a minute. You see, now your resin is having your DNA. We have again charged the cartridge, and we are washing it. Now, during the washing step, your DNA is still sticking to the resin. It is still in the resin. It is just washed to remove the salts and other. Now you see, this wash has been poured back. Again, the cartridge is added. This washing can be repeated 3-4 times. Again, centrifuge, so that your resin gets washed, which is actually having your DNA. Now you take out the cartridge, throw away this discard. Now the final step is to leave it for 1 minute and place at room temperature for 10 minutes. Then you add the elution buffer. Now whatever was bound to your resin will be removed. This is the addition of elution buffer. Now elution buffer is nothing but 1 molar NaCl, which will actually disconnect the connection between the charged resin and the charged nucleic acid. So this buffer, once you put this buffer over the resin, it disconnects the nucleic acid with the resin. And when you give a spin, your DNA is going to come down at the place where previously discard was coming. Now, once you spin this with the elution buffer added, the spin is carried out for 5 minutes. And now what you have below is your DNA or the plasmid. So this is how you purify your plasmid DNA. So that was mini prep. Plasmid, as you know, is a vector, is basic to every process of cloning a gene. So nowadays, as a time of automation, we don't do the classical methods. We are using these kits. But I have also told you the theory behind the isolation of plasmid DNA. I think you can now understand the principle behind separation of plasmid DNA. So now